Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on Punnett Square. So today in this lecture, we are going to discuss about the Punnett Square, how the Punnett Square is made, how the Punnett Square is beneficial, what are the uses of Punnett Square. All this thing we are going to discuss. Okay, so we are starting with the first point. So first of all, see, this is the title of today's lecture. Okay, a Punnett square can be used to predict the outcome of crosses and self-fertilization experiment. Okay, now what is Punnett square? So Punnett square is an easy way to predict the outcome of simple genetic crosses and self-fertilization experiment is to use a Punnett square. Now, now first of all, let's understand the Punnett square. Now, you and me, both of us, are not Mendel, and we are not performing our experiments on the pea plants. Even though someone tell us that you find out the progeny, you find out the offspring's genotype and phenotype. Let's say someone tell us that you start the cross with the tall heterozygous and dwarf homozygous plant. Okay, so what is the question? Someone is telling us that you start with the heterozygous tall plant and homozygous dwarf plant, and what will be the genotype and phenotype of the offspring. So this we need to find out. Or someone tells us that you perform a cross. Okay, you tell us the result, between the heterozygous purple flower plant and homozygous white color flower plant. So what will be the offspring's genotype and phenotype? So now with the help of Punnett square, we will be in a position to find out the genotype and the phenotype of the offspring. Okay, so let's find out how this is done. First of all, who developed this method? Okay, so a method originally proposed by this method was developed by British geneticist Reginald Punnett. Okay, Reginald Punnett is the scientist who developed the Punnett square. Now, to make the Punnett square, to construct the Punnett square, first of all, you must know the genotype of the parent. So, if we want to make a Punnett square, we must be knowing the genotype of the parent. So, the experiment from where we are starting, the first step, the first requirement for us is to know the genotype. The parent, that means the parent is homozygous or heterozygous. Okay, now with this information, the Punnett square enable you to predict the type of offspring the parents are expected to produce and in what proportion. So once you are knowing the genotype of the parent and you make a Punnett square, and then with the help of Punnett square you can predict, you can calculate the genotypic ratio and the phenotype ratio for a particular character or the trait. Okay, so this is the usefulness of the Punnett square, that you can know, you can find out the genotypic and the phenotypic ratio of the offspring. Okay, now these steps, okay, how to make the Punnett square. So this is the step number 1. Okay, over here it is written. See, write down the genotype of both the parent. The first step is the writing the genotypes. Okay, first you write the genotype of both the parent. Now, in a self-fertilization experiment, a single parent provides the sperm and egg cell. We know in the pea plant flower, the natural tendency for reproduction is self-fertilization. And in self-fertilization, the single flower is providing both the pollen and the egg. Okay, that means in the pollen we know the male gamete or the sperm is present. And in the ovule the egg cell is present. So in the self fertilization, the single parent is providing the sperm and the egg, or the male and female gamete. Now, now let us consider an example in which heterozygous tall plant is crossed to another heterozygous tall plant. Now to develop the Punnett square, we are starting with the heterozygous tall plant. And heterozygous tall plant, we know heterozygous tall plant is having one dominant allele and one recessive allele. Okay, that means one allele is capital T, another allele is small t. So now what we are going to do, we are cross fertilizing. That means we are transferring the pollen from one flower to the stigma of another flower. And here in this case both the plants are heterozygous. That means the allelic combination they are having is capital T small t. Okay, now the plant providing the sperm is the male, okay, parent. The one which is providing the sperm is considered as the male parent, and the plant providing the egg is considered as female parent. Okay, so the genotype of male parent is capital T small t, and the genotype of female parent is also capital T small t. So both the parents are heterozygous in this case. Okay, now the step number 2. See, step number 2, write down the possible gamete that each parent can make. Okay, so the second step, we are writing we are writing the possible gamete that each parent can make. Okay, now remember that the law of segregation tells us a gamete carries only one copy of each gene. So in our previous two lecture we have discussed this law of segregation, and law of segregation tells us that when the gametes are formed, only one allele is present, or we can say the alleles are separating at the time of gamete formation. So each gamete receives only one allele, one copy of gene for a particular gene. Okay, so in the male gamete, either capital T or the small t allele will be present. Okay, so let's say the half the male gamete are having capital T, and another half male gamete are having small t allele. Similarly, in case of female, okay, in case of female gamete, there will be, in the female gamete, half the female gamete they are having, let's say, capital T allele, and half they are having small t allele. Okay, this is the step number 2. Okay, see this entire lecture, okay, on Punnett Square I have uploaded on my app. So to watch this full lecture, I am inviting you on my app. Till then, bye bye, namaste.